अच्छा चले you can see the screen right? तो अफान आवाज आ रही है?
यस सर, आवाज आ रही है।
जी। अच्छा, so remember we just did transition metals and we did the whole thing के एक second, let me just open the syllabus ताकि I can, where's the syllabus? Just one second. I think it's 2025 2027. ठीक है, give me one second. अच्छा, इसमें where is, yeah, why don't I get transition? Just one second. अच्छा नहीं, आई थिंक मैंने ओ लेवल्स का खोल लिया। जस्ट वन सेकेंड। It is, yes. Why don't I get transition metals? Hold on one sec. ठीक है, I think it's before this. Colors of complexes. Yeah, this one. Though we were doing all of this, we did, yeah, we did the monodentate complexes. और इसमें we did water NH3Cl, not Cl minus one yet. वो हम कर लेते हैं. We've done, yeah, we done copper 2 and cobalt, right? Their complexes and their colors, etc. So one by one, the things that are left, I'm going to move towards bidentate ligands, ठीक है? So types of ligands now. So ये वाला first part is this clear that you've got remember copper and cobalt 2 complexes. Those are the only ones that are in the syllabus, not the other ones. So this was the last thing that we did. अब इससे आगे ये कि you've got types of complexes, right? So the first thing is types of ligands. Let's start with types of ligands. You've got monodentate ligands. ठीक है, that's मतलब ये फर्दर उसकी क्लासिफिकेशन वगैरह, ठीक है. सो मोनोडेंटेट लाइगेंड्स इज कि it's a ligand that can only form one dative bond. That's it. So it's a ligand which forms exactly one dative bond. So all these ligands that we studied, they were all monodentate because they would only form one dative bond. Cl minus one water, that's a monodentate ligand. You've got NH3. ठीक है, that is also a monodentate ligand. Or syllabus with this, they've also written Cn minus one, so that's also a monodentate ligand. ठीक है. So that's what a monodentate ligand is. But is that clear? It only forms one dative bond. Is that part clear?
यस।
I said then you have other ligands which are bidentate ligands. Dentate means teeth, right? So bidentate ligands have two lone pairs and they are capable of forming two dative bonds. So those are your एक second है ना. अच्छा, so those happen to be your, अच्छा, those happen to be your bidentate ligands. So ligands which form two dative bond. With what? With a central metal cation in a complex. Now, for this, you have to remember two specific reactions, two specific bidentate ligands. One of the, and I'm going to show you how it forms two dative bonds. So one of them is a diamine. It's ethyl diamine. So it's CH2 CH2, and it's got NH2 on both sides. So it's got two lone pairs, and that's ethyl diamine or ethane diamine. The other one is ethane dioic acid. It's or the ion O minus one. This is double O and O. Now this thing also has, so that's your ethane di O8 ion. ठीक है. So these two ligands, they form they form two dative bonds, and I'm going to show you how. So for example, I've got iron. It gets just as needs crash. Just 1 second. Yep, this one. ठीक है, सॉरी वो क्रैश हो गया था. अच्छा, so you've got इन दोनों के नाम. I'm going to write the names again. अच्छा, so this one is ethane di-O8, and that one is ethylamine, diamine, or ethane diamine. अच्छा, so the thing is, how do they form two dative bonds? So you'll be asked to draw these. So for example, I've got iron 2 plus complex, right? And the iron 2 plus has ethane diamine attached to it. So since it's got ethane diamine that's going to be attached to it, so that's one. ठीक है, one ligand would be attached at the bottom. ठीक है, there's going to be one that's going to be, so that one is going to be going into the page or into the board. ठीक है, there's going to be another one that's going into the board. इसको एक सेकंड सही कर लें. Yep, that's fine. And this one's supposed to be a dotted line that just shows kids. So that shows it's going into the board. And then there'll be two of them that will be coming out of the page. ठीक है, so let me just. ठीक है, so that's one. So that's going to be your wedge-shaped bond. And you're going to have these on the other side as well. So this is it. So one is wedge-shaped that's coming towards you. So that those are your X, Y, and Z axis. Anyway, so you're going to draw, and I'm going to draw both bidentate ligands attaching to this iron 2 plus complex. Okay, so one of them is over here. So what's going to happen is, okay, the first one, let's look at ethane diamine. This one over here. Remember, it's got two NH2 groups, and the NH2 groups have lone pairs. So the way it's going to get attached is, okay, there's going to be N, H2. Okay, think of it as two ammonias. It's got NH2 on this side, and there is a carbon chain, CH2, CH2 in the middle. So one end of the ligand gets attached, and it's lone pairs. The N lone pairs get datively bond to the Fe2 plus from one side, and the other one they datively bond to the Fe2 plus from the other side. Okay, is this part clear? How bidentate ligands attached to the iron 2 plus in the complex? Is this clear?
Yes.
You get the same thing over here. The NH2, there's going to be another NH2, and you're going to have CH2 and CH2. Okay. So this, and the lone pair is getting attached from one side, and the lone pair is getting attached from the other side. Same thing over here that there is going to be NH2, okay, and there's going to be two CH2 groups in the middle, and the N lone pair is getting attached from two different sides. So that's going to be your entire complex. So that's the way I'm going to write this is Fe. Now they could give you another bidentate ligand. You're going to do exactly the same thing. So it's Fe with what? With the diamine, which is NH2, CH2, CH2, and then there is another NH2 thrice. And the overall charge on this entire complex is going to be +2 because the diamine has no charge. There is no charge on the diamine. So the whole entire thing will have a charge of, it's going to have a charge of +2. So you just go, we can, so we can write the charge outside it. Okay. So the overall charge on the entire thing would be, is going to be +2. Okay. So is this clear how to draw a bidentate ligand? Clear?
Yes, miss.
Yes, sir.
As the same with the other one, ethane diovate. You're going to do exactly the same thing. So ethane diovate, the complex would form, and you will have, you've got O, and you've got C. That's a double bond O, double bond O. This side is -1. This side is -1. So that's one ethane diode getting attached to it. Then you have another one, और ये बीच में ना there's C and C double bond O, and then there is, here that's going to be your ethane diode complex. And if you write it together, it's going to be what? It's going to be Fe with C2O4, or you can write the whole thing, or you can just compress it. It's C2O4. It's got a -2 charge. There are three of them. So the overall charge, if Fe is 2 plus and C2O4 is has got a -2 charge, so what would be the overall charge? That's going to be क्या होगा? That's -2 times -6. So the overall charge on the entire complex would be -4. ठीक है? Is this part clear?
Yes, sir.
अच्छा, फिर next is that you've got so now next is that you've got so that's it, that's bidentate ligand. And then there's a polydentate ligand as well. So what's a polydentate ligand that you could, it could form many dative bonds. Okay, so polydentate ligand is, Now, polydentate ligands are normally,
Sir,
yes.
how come for ethane dioate the charge is -4 and not for
You see,
this ethane diamine?
what is the charge on C2O4? That's -2, right?
Yeah.
What's the charge on Fe2 plus? That's so you collect all the charges, right? It's this is +2.
2, so then it cancels out, and then you're saying that you have extra 2.
Yeah.
So that becomes -4.
So
Okay, got
that's
it.
why over here it's the diamine was neutral, right? So it didn't have a
Mm,
charge, and it's Fe2 plus. So 0 plus Fe2 plus 0, it adds up to +2, right?
yeah.
Okay, so remember you'll be asked to draw this. So that's important. The drawing is important. Then you have polydentate ligand. Now that's very complex. You're not going to be asked to draw this. I mean, you can be asked to draw this, but normally, I mean, the drawing would be really difficult. Polydentate ligand is a ligand that forms many dative bonds. So a single ligand can form many, many dative bonds, just like a bidentate. So it forms many dative bonds. Poly means many. Now there's one molecule, you should remember the molecule. Well, you can't, it's ETA, EDTA. It's got a complicated name. I'll just post a picture of that. You can't remember the molecule. You can only remember the name of the molecule because it's like a complicated molecule. So ETA ligand is like this. It's the name is ethylene diamine tetraacetic acid. So forget the name. Just remember ETA. That's the short name. The molecule looks like this. So it's this large molecule over here. So I'm just going to copy this image. I mean, if you see the picture, you should know that that's it. You possibly can't remember this molecule, the entire molecule. Uh, but if you see a picture, they'll just simply use the word ETA. That's it. They won't even draw this. So ETA is this thing. It's a big molecule. It's a large molecule. What it does is that it wraps around a metal ion and starts forming dative bonds from all sides. And specifically not this. This is not the one. Just one second. That's not the one. The ion. Where's the ion? Just one second. Yeah, this one. I mean, the carboxylic acids would lose the H. I'm just going to correct it over there.
Is the maximum amount of dative bonds that can be formed with a metal ion 6?
Normally, it is 6 because we run out of space, basically.
But
So
with
if you
this, how many would it be?
6,
6 only?
yeah.
Okay.
So if you're not running out of space, I said, "Okay, just give me 1 second. Let me just put this thing on charger. Okay, just 1 second. Okay, so this one. Can you hear me? Okay. Can you guys hear me?
Yes, sir.
Okay, so I'm just
Yes, sir.
remove. I'm just the carboxylic acid loses the H plus one, so that's your. So that O has one lone pair, right? This O over here lost an H plus one, so it has. These are all carboxylic acid groups, right? So it loses an H plus one. This one also loses an H plus one, right? So in total, this one has a lot of lone pairs. It's got a lone pair over here. It's got a lone pair over here. It's got a lone pair over here. So it's also going to form 6 dative bonds. So it's kind of going to wrap around a metal ion and will start forming dative bonds from all sides. Okay, there was an image given over here. Let me show you the first one. So this is how your eta molecule would. You get to this one. So that's how your eta molecule or ligand or ion would wrap around from all sides. I mean, the oxens will start forming dative bonds from the top. Someone would be coming from the right side. Because it's a large molecule, it will completely wrap around the metal and start forming a lot of dative bonds. Normally, it's going to form six as well. ठीक है, is this clear that what a polydentate ligand looks like?
Yes, sir.
ठीक है. Also remember,
Yes, sir.
most natural molecules, like you've got metals in biology, right? Like we've got metals in our body, right? For example, there's iron in the hemoglobin. So all these are forming complexes like this. I mean, this is what a heme group looks like. It's like an iron that's trapped. Um, One second, not co-pilot. So it's like an iron that's trapped. There's a metal ion that's stuck between inside it. So for example, this one. I want a 3D diagram. Just one second. If we have, yeah, this one is the one. So the heme group is kind of like this. It's like a polydentate ligand. So what happens is that in your heme group there's a complicated molecule, like a really complicated molecule. That's your heme group, and there's N with lone pairs. Right in the center, there are nitrogen atoms, and those nitrogen atoms have these lone pairs. And there's an iron 2 plus ion that's stuck in the middle. Okay, so that's how hemoglobin gets formed. And there's an N lone pair that's coming from the bottom as well, right?
So what's the benefit of this? Does it like make strong structures and stuff?
The thing is that the role that the iron plays in your body, in your blood, in your red blood cells is that it has one side that's open. So what happens is that's the oxygen binding side. What happens if there's an oxygen molecule coming in, that oxygen molecule has lone pairs as well, right? So it ends up getting attracted to the iron 2 plus from one side. So it's like all the other
Does it
place.
does it
Yeah,
form a dative bond with the iron as well, the
yeah,
oxygen?
exactly.
Oh, okay.
So that's how the oxygen gets absorbed into your red blood cells, right? Is that clear? So it's like a complicated molecule. It's a polydentate ligand with an iron ion that's stuck in the middle, right? With one side open. And the thing is that the oxygen dative bond is not very strong. The other bonds are like relatively stronger. The oxygen lone pair is getting attracted to the iron 2. That's not a very strong bond. So this bond breaks easily as well. So oxygen gets absorbed and it gets deabsorbed as well. Is that point clear?
Yes, sir.
And there's another uh, thing apart from oxygen molecules, but they form a very strong bond with that iron ion, and that's carbon monoxide. So if you have a carbon monoxide molecule coming in, so that carbon monoxide molecule, it also has lone pairs, right? That can also form a dative bond with the iron ions. But the thing with carbon,
So, sorry, like it is strong because it had three bonds, so it makes stronger bonds with iron.
because it's more polar. Because carbon and oxygen molecules are
Oh,
not polar. This time, what is happening is that the oxygen is more electronegative, so the electrons are closer to. So this side is more negative, right? So which is why the attraction, because carbon monoxide is a polar molecule, so the attraction between the iron 2 ions and the carbon monoxide is a lot stronger. Okay, is that clear? So
yes, sir.
remember, you're not going to get any specific question on this. You would probably get a question similar to this, but they're going to pretty much explain you everything. You're not going to draw this. You're not going to show the data for the drawing part. You'll probably 99% chances are you're going to get very simple questions. That's it. Okay, so you're not going to get you're never going to get drawings for polydentate ligands. I mean, that's because it's impossible to draw them. Okay, is that clear? So
Yes.
so the thing with carbon, so you can think of this as like all the information would be given to you, and they'll be asking bits and pieces of information about this structure. Probably not going to ask you about the drawing. As a carbon monoxide, remember it's going to form a very very strong dative bond, which is why the iron gets is no longer able to carry oxygen after that because it's going to permanently bond with the iron two ions. Is that clear? So that is why carbon monoxide causes choking and it reduces your oxygen in your blood and eventually you faint and you don't even realize that it's a very very silent killer. ठीक है? Is this clear?
Yes, sir.
That's the next one. So that's all polydentate ligands. So remember, there are three types of ligands. Bidentate, you'll be asked to specifically draw these two, or maybe some other molecule which is a bidentate ligand. Polydentate, not going to be asked to draw this, but you should know what a polydentate is. The other thing is, okay, we can discuss isomers as well. So you've got isomers and complex ions. Now the isomers are kind of the same as the isomers in you're going to have cis trans isomers over here as well. So the first one, they're known as geometric isomers. So geometric isomers are your cis and trans isomers. Now when you have geometric isomers, what happens in geometric isomers is, well, you've got same side, if you remember organic chemistry, same side and opposite side. So the first one I'm going to do is in square planar, sorry, not in square planar, in octahedral complexes, the one that we studied. So you will have geometric isomers in octahedral complexes. For example, we had done this molecule which was CuNH3 4 and H2O twice, right? And it had a charge of +2. This was the deep blue solution that we studied earlier on. Okay, you had water, and the water was coming in. Sorry, the ammonia was coming in. Just a second. So you had, in this one, you had ammonia. So we did do this one. So I'm just going to pick this. You're a picker. I'm just going to make adjustments on top of that. Okay, so let me get rid of. So forget the iron ions. Okay, this one. And again, the drawings are important. You'll be asked to draw this. So how do four NH3s and two water molecules get attached to it? Now there are two ways. So there's copper 2 plus in the middle, and there are going to be four NH3s that would be attached to the copper 2 plus. So here are your four NH3s with their lone pairs. There's another NH3 with its. And there are going to be two water molecules that are going to be attached to it. So in the remaining two spots, you got two water molecules. So that's your copper with the, so that's the complex. You clear? Is this part clear? Zohair, Zaina, is this clear? Amna, is this clear?
Yes, sir.
Okay, so that one is the first one, and the overall charge is +2. Now this one, if you see carefully, the water molecules are opposite to each other. They are 180 degrees apart. So opposite side, that's your transversion. Okay, if you remember organic, trans was when the same type of groups were on opposite sides, right? And then you have another one which is going to be your cis version. In your cis version, the water molecules will be on the same side. So the water molecules. So I'm gonna just switch positions. The water is not here. The NH3 is not. Okay, let's replace this NH3. So the water molecules are like next to each other, and the NH3s are covering the other spots. So now what's happened is that the water molecules are 90 degrees apart, and that's going to be my cis version. Is this clear?
Yes, sir.
Yes, sir.
Okay, so Cu NH4 H2O twice, it's got two versions. One in which the water are like opposite to each other, and the other one where the water molecules are next to each other. And then there's another one. You could also have octahedral. You could also have isomerism when you have this thing. So I mean the same molecule. There could be another one. What if you have three NH3s and two water molecules? So if you have three NH3s and three water molecules, even then you can have three different arrangements. So if you have three NH3s and three water molecules, this is one. That on this spot I'm going to have a water molecule. So that's so three water, three NH3s, right? Or there could be another version where you could have uh and let me draw the second version. So I'm putting the water molecule on this side. So there are two different arrangements. Now remember, the arrangement is based on the bond angles. Like over here, if you look at this arrangement, these three, I mean, you could have rotated versions as well. So over here, what happens is that between the water molecules, there are 90-degree bond angles, right? I mean, this water over here, that's 90 degrees, this water over here. But between two water molecules, the angle is 180 degrees as well, right? So over here in this arrangement, they're making both angles. They're making a 180-degree angle between them, and they're also making 90-degree angles between them as well. Is that clear?
Yes, sir.
And in this arrangement, none of them are making 180-degree angles between themselves. They're all close to each other. Over there, they were like spread out, they're far apart, they're on opposite sides. But over here, they're all on the same side. They're all pointing in the sort of facing the downward direction. So they're all making angles of 90 degrees only. Okay, so that's your other version. So remember, uh, you're going to face cis-trans isomerism or geometric isomerism when you have either four ligands of one type and two ligands of a different type. So you can get these two combinations. Or you're going to face geometric isomerism if you have three ligands of one type and three ligands of another type. So the three could be like on opposite sides or they could be all together right next to each other. Okay. Now,
Sir, can you say that in a sentence so we can write that down?
I'm going to tell you that so what I'm going to write is that let's say you've got CuNH3, X and H2O Y. So X and Y ratio, right? So if it's either 2:4, like four of one and two of the other, or if it's 3:3, in both cases you're going to have geometric isomerism. Okay, the ligands could be distributed the way I've showed you. You're not going to have geometric isomerism if you have what's the other combination? Like the total is six, right? Because it's an octahedral complex. So what am I left with? I'm just left with one and five, right? In one and five, you're not going to get geometric isomerism. Over here, you're going to get geometric isomerism. Like in 1 and 5, like if you just had one water molecule, then obviously there's no other version of that. Okay? And the thing that you have to be careful about is the rotational thing that, I mean, remember this thing over here? You could be given a rotated version over here. The water could be on this side and the water could be on this side, right? But remember, you're just going to look at the angles, right? That they're opposite sides. So that is still going to be called trans. The same thing over here. The cis version would be that the water molecules are like next to each other. So they could be next to each other. I mean, this water could be, you could be given a rotated version as well. I mean, the water could be on next to each other on this side as well, or they could be next to each other on this side, or they could be next to each other over here as well, right? So it doesn't really matter as long as they're next to each other. That's going to be the cis version. Okay, is this clear?
Yes, sir.
Okay, same thing over here as well with 3 and 3. The water molecules right next to each other, all making 90 degrees. So they could be on the top side, they could be on the bottom side, they could be over here, water over here, water over here, water over here, as long as they're all 90 degrees next to each other. And this thing over here as well, like you could have a rotated version as well. So that's octahedral complex, and that's the geometric isomerism in octahedral complex. Is that part clear?
Yes.
So there's another complex which shows geometric isomerism, and that's so that was part one, octahedral complex. So there's another one which is part two, and those are square planar complexes. Now we did not draw any square planar complexes yet. The only ones we drew were the tetrahedral ones and the octahedral ones. So you've got square planar complexes as well, and there's only one you have to remember, and that's known as cisplatin and transplatin. So hopefully you know what the shape is, square planar. It's got there's a platinum ion. Square planar is literally square. It's you've got you just have four bonds, and they are all 90 degrees to each other. Okay, so there's a platinum in the middle, and here are your four bonds, and the shape of the molecule is exactly 90 degrees. It's completely flat. Okay, it's like think of the page, and there's no bond coming towards you or going into the page. It's completely flat. All the bond angles they're all 90 degrees. Now, cisplatin is a molecule. It's a very important molecule, by the way. I'm going to tell you what the importance is. It's got two NH3s bonded to it, and it's got two Cl's bonded to it as well. The platinum in the middle is +2. So it's got two Cl's bonded to it as well. Now the Cl's are on the same side. I mean they're close to each other. So this one is the one that's cisplatin. Okay, that's your cisplatin. And then there's obviously another one which is transplatin. It's the same shape. Okay, and since it's got the same shape, the only difference is that the NH3 and the Cl's are opposite to each other. So the Cl is over here and the NH3 is on this side. इसको सही करके draw कर लो. 1 second. So the Cl is at the top and the NH3 is over here, and the Cl's are like opposite to each other. So that's my transplatin. Okay, is this clear? This thing.
Yes.
Okay, so you got to remember square planar complexes like cisplatin and transplatin, they're going to have geometric isomerism as well. So this is your cis and opposite side, so that's your trans. Okay. So now, is this clear to everyone, Zaira? Is this clear? Akshita, is this clear? Maheen?
Yes, sir.
Achha, now the importance of this thing is that it's a very important. I mean, this one, cisplatin. Cisplatin happens to be a very important anti-cancer drug. Now the problem is that remember the shape of the molecule, especially in biological molecules, it's very important because you've got active sites, and the molecule has to fit in the right places, right? Otherwise, the molecule will not be able to react with the biological because like we saw the heme group, right? Biological molecules are like really complex, and they've got like a geometry that has to be taken or considered, right? So the so whenever you have a drug molecule or an enzyme or anything, it has to have the right shape. So the transplatin would probably not work, and it would probably cause side effects because it will work in places where you didn't think it's going to work. Uh, So maybe if you it's like cisplatin is an anti-cancer drug, it attacks cancer cells. Transplatin might attack some of the healthy cells. So the problem with synthesizing these molecules is that the geometry of the molecule has to be considered as well, because one of them could have side effects and the other one could be doing a wonderful job, right? So this one is the good one. The other one we don't want. Now, how does and again, this one is just a general idea. You'll be asked to draw this. And you should know a general idea how cisplatin would work as an anti-cancer drug. So I'm just going to briefly tell you, just in 4 minutes, I'm just going to briefly tell you what cancer is. It's basically your DNA that gets damaged, right? And the cells they start replicating, and the DNA contains all the information about protein synthesis. So what happens is that when such cells which are damaged, which have damaged DNA, they start replicating. The problem becomes that those cells are useless. So they cause or they produce tumor. And those tumor cells, they start producing proteins, which are also not, I mean, they're all damaged as well because the DNA code is damaged. So the tumor multiplies and grows and it spreads and it has to be stopped. So you have to kill those cells. So the way this is done is, and the drug delivery has to be very targeted because you don't want to kill the healthy cells. So if somebody has brain cancer, you just need to, the molecule has to attack the cancer cells only and not the healthy cells. Okay? Because if you kill the healthy cells, then that's basically game over. Okay? You don't want to kill the healthy cells. So what happens is that DNA is like, I don't know if you're taking bio or not, but DNA is something like this. It's like a polymer chain. It's got a phosphate group, it's got a sugar molecule, and it's got a nitrogen base. And if it's got a nitrogen base, nitrogen has lone pairs, right? So the DNA is like a polymer, again, sugar molecule, which is connected to a nitrogen base, and the strand continues, right? You got phosphate group, sugar group, and there's an there's a nitrogen base which has lone pairs. And remember, it's like a complicated molecule. I'm just writing S, N, and P, but nitrogen base means it's like this is what the actual DNA molecule looks like. If you want to look at the whole thing, it's so the DNA structure is something like this, not this one. Yeah, this one. So this is how the DNA is. It's that's your sugar group, that's your phosphate PO4 group, and that's your base. It's got a couple of nitrogens in it. So that's your nitrogen base. That has lone pairs in it. And the DNA strand is connected with another DNA strand. It's like a ladder-like structure. So there's a complementary DNA strand that's on the other side. So there's a sugar molecule, there's an N nitrogen base, a phosphate group, then a sugar molecule. So there's a complementary strand. So it's like a ladder shaped. If you haven't studied bio, it's you've seen it in a number of places, a spiral structure with a ladder shaped structure. And it's got four different types of nitrogen bases, TK, which are guanine, cytosine, adenine, and thymine. So very quickly. Now in the cell, this ladder-like structure, they are hydrogen bonded together. There's NH groups over here on both sides, and they start forming hydrogen bonds and they start sticking together. So it's like a ladder which is hydrogen bonded or glued together. Is that clear?
Yes, sir.
So when the cell divides, the DNA strand also divides. One DNA strand goes into one part of the cell, the other one goes into the other part of the cell, and the whole DNA strand gets copied again from that partial DNA. ठीक है, is that clear? That like the two strands and they unzip and they create uh two copies of themselves. Is that clear?
Yes, sir.
So if the DNA is damaged, you want the DNA not to replicate. You don't want it to unzip. You don't want it to split. You want to prevent cell division, right? So the way cisplatin does this is that since it's got these there's ligand exchange that happens, the platinum comes in. I mean, there's Cl-1 that was already attached to it. And you had ammonia, right? The ammonia ligands get displaced and they get replaced by the nitrogen bases. Is that clear? कि वो जो square planar cisplatin है ना, that comes in, and the ammonia ligands they get displaced and it gets attached to the nitrogen bases, and that keeps the two strands together. Is this clear?
Yes.
So that prevents cell replication and cell division because now the DNA will not be able to split, and if it's not able to split, it will not be able to copy itself and create further copies of itself. ठीक है? Is that part clear? Just very general information. You might be asked to explain this, but not in very detail. It's just going to be very, very general. ठीक है? Is this part clear to everyone?
Yes.
ठीक है, Zohair? Is this clear? Amna? Akshita, is it clear?
Yes, sir.
Yes,
Yes.
sir.
So, ठीक है. So let's continue next time. We still have some isomers left. ठीक है? So we'll continue tomorrow. Okay then. Take care.